So, guys, we have seen that transamination leads to the transfer of amino group of an amino acid to a keto acid to form the glutamate. So, of course, it gives rise to the formation of a glutamate, but we do not get rid of amine group or ammonia, but the main one of the major part of the catabolism of amino acid is to get rid of the ammonia. So, what for that there is a second process as far as the catabolism of amino acid is concerned, that is what we say is the oxidative deamination, because it leads to the formation of NADH2. So, if I can show you here that this is of course the glutamate, let us say you get it from the transamination of any other amino acid, and this glutamate then under the action of glutamate dehydrogenase, it gives you the alpha ketoglutarate, fine, because of the and as a result of this to get the ammonia out, and this ammonia then is excreted from the body through urea cycle in the form of urea. Also, as I have told you that because the NADPH actually because NADH is formed, it means that it, of course, we have been seeing here that NADH in the mitochondria gives rise to the formation of ATP. So, it is a energy giving reaction also. The enzyme, of course, here is the glutamate dehydrogenase, and the coenzymes are NAD and NADPH. Of course, where NAD is for the oxidative deamination basically, and NADPH is for the reverse, that is for the reductive amination. As far as the regulation of this reaction is concerned, of course, it is a general sort of regulation which depends upon the amount of reactants or products you have, and also on other certain cofactors. For instance, if there is more or you have more glutamate, it will give rise to more deamination. If you have more ammonia, it will lead to inhibition of deamination process, and also then GTP leads to the inhibition of this enzyme, that is glutamate dehydrogenase, and the what we say the ATP leads to the activation of this enzyme. In addition to these regular type of amino acids, there are also D amino acids which are acted upon by the D amino acid oxidases. For instance, the D serine is, of course, it does not become the part of these D amino acids, that do not become the part of our proteins, but still they have some clinical implication. If some defects occur in these in the metabolism of these amino acids, it have some clinical implications also. For instance, if there is failure in the metabolism of D serine, because the D amino acid oxidases are not able to really metabolize or degrade this amino acid, it will lead to certain neurological disorders like the schizophrenia. So, of course, one of the benefit of oxidative deamination is, of course, that you get rid of the amine group by getting it out of the glutamate, but then also you should keep in mind that transamination and oxidative deamination also make the ground for making new amino acids, because once you get rid of an amine group from glutamate, or once the transamination takes place, you are then ended up with new acid groups which can be then used for the process of what we say the reductive amination. These reductive amination will then lead to the formation of new amino acids, because here what will happen that depending upon that ammonia will bind with which acid group, it will give rise to the specific amino acid. So, in the reductive amination of course require, because it is a reductive step, it requires NADPH, and as a result of this it is an energy intensive reaction. So, ammonia is used in this reaction, and as a result of this what will happen that ammonia along with alpha ketoglutarate give rise to the formation of glutamate. So, this is one of the amino acid which is formed through this process. Similarly, you can get alanine, you get serine, you can get other amino acids through this aspartate, you can get other amino acids through this process. In this way, transamination and the deamination, not only, or oxidative deamination more accurately, not only serve as a way to get rid of ammonia or to transfer ammonia from one amino acid to the other amino acid, but it also paved the way for formation of all sorts of amino acids also. In this way, it acts as a very hub, central role. It plays central role in the metabolism of amino acid, whether it's about the biosynthesis of different amino acids or whether it's about the degradation of different amino acids. Now, the ammonia released after the oxidative deamination, it will be, because it's a very toxic compound, it has to, body have to get rid of it, and we will see how urea cycle help in it. Thank you.